Do you have any idea that when your kidneys get sick, the microbial world in your saliva also changes? So my work is related to that. I work with the salivary microbiome, which means that it's the microbes which are living in your saliva. So we call it microbiome because it's like the DNAs of these microbes, which we look into for our studies. And the research work is dependent on one kidney disease specifically. It is called Berger's disease or IgA nephropathy. And this disease is kind of the leading cause of chronic kidney disease around the world, and especially in a country like Japan. So Japan is second highest in prevalence, and it also has a very rapidly aging population. So the confirmatory test for this disease is kidney biopsy. But kidney biopsy is a very high risk diagnostic tool. And also for aging population, it's a greater risk at that. So we need a tool which is as effective as a biopsy tool, but it's not as high risk or as invasive as this tool. So that's where we look into salivary microbiome. So why would you think about salivary microbiome to connect with kidney? That was the first question that comes up in everybody's mind. Is it's because of the two main reasons. First is saliva is very easy to collect. Like anybody, you can collect. You don't need a doctor's appointment for that. You don't need an instrument for that. You just need a tube that the doctor will give you, and you can spit into it, and you just have to send it out. So you don't even have to come to the clinic for that. You can just post it. And the second thing is it's these days, like there have been a lot of research around saliva and the disease that happens all over our body, not just the teeth or mouth diseases. It can be anything. It can be it can be a disease in your gut. Like if you have ulcers in your gut, then that changes how your saliva looks. Or if you have arthritis, that also changes how your saliva looks. So that's how we thought, okay, if it's connected with everywhere else in our body, maybe it's also connected with how our kidneys function. So that's how it started. And so what we did is we collected saliva from healthy individuals, which were just individuals who came into hospital for their health checkups, or they had matched all the parameters where we see them as healthy. And then we had patients who were diagnosed for IgA nephropathy, and they had come to the hospital for their surgery of tonsil removal. So in Japan, if you are in your early stage of the disease, the best therapy for you, they will remove the tonsils and they'll give you the steroids for the treatment. So that's when we collected their salivary samples. And so this was the starting point. And so now we have the saliva in the tubes. Now what we will do is we will do sequencing. So sequencing is we will extract all the DNA that was there in the saliva, and we will selectively choose only the microbial DNA that does only belong to the microbes, and we will sequence them. And sequencing means we are just taking snapshot, like we are taking pictures of that saliva to see who are present in your saliva, like which microbes are more, which microbes are less, or who are just there, who are just hanging out in our saliva. We'll just get an idea about that. And we want to also use this information to see if we can separate a healthy individual from a patient only using this information. So this was our study design, and we moved on from here. So what we found out was indeed the salivary microbiome of the IgA nephropathy patients significantly changed as compared to the healthy individuals. And in our technical term, we'll call this dysbiosis. So dysbiosis means just an imbalance in how the picture looks. So if a healthy picture looks some way, you can see that the IgA nephropathy picture will look different to that. We can't say if it's better or for worse, but it has changed. So that is important. There is a change. And we define this change in two terms, that is richness and diversity. So richness is, which means that how many of these individual microorganisms are present, like how much of them are present in the sample. That's the richness. And diversity is like, is a usual dictionary meaning, that how evenly are they distributed? Are they all of the similar proportions, or are they somebody very high or somebody very less? So that's how the diversity works. So in terms of richness and diversity, also it was decreased in case of IgA nephropathy than the healthy individuals. So now we know there is a change. So how does this information help us to classify? So we use that so nowadays we have this machine learning tools. So it's just computer programs where you can separate, you can see if you can separate two groups based on some parameters. So in our case, parameters was the quantity of the microbes. And our two groups are the healthy and the IgA. So we tried to separate them, and we saw that a group of about 9 or 10 microbes were able to separate the IgA nephropathy group from the healthy group with about 90% accuracy. So that works. And if we have, so now the future prospects of this group will be if we can add some other medical parameters like kidney filtration rate or any other parameters that the physicians usually test for the patient. If we can have that information in the model, it will just make the model perform better and become more efficient, and we can use that as a diagnosis tool for the disease. So this will make much easier for the patient, also for the doctors, because you don't need any other instrument for this test. And that's the importance of this work. So that's all I have to talk about. So thank you for listening.
Cool. I never knew there was so much information stored in saliva.
So even for a longest time, saliva was only connected with the oral diseases. So nobody thought about how they can change depending on anywhere else in our body. But it seems that the microbes especially are very sensitive about what goes on in our body, because they are like our best friends. So whatever happens to us, it kind of like, it does knock them off. So they are not happy with if it's not a happy change. So they will change according to whatever happens in our body. And that's how they are changing also in our saliva, even if we have a kidney disease, which is like, yeah, they are very far apart.
Right. Yeah, that's so cool. I would imagine doctors in hospitals are really interested in this sort of thing. Is that?
Yeah, so microbiome as a whole is kind of, it started with that initiative that we want to connect the research with the medical world. So like the application of this field is medical world, or not just in terms of therapies, but also for like prevention of disease. So if you know how healthy microbes look like, or how a healthy system looks like, then you can tell them how to attain that healthy system. So that's how the application is there. And also like this study, it started with that motivation that this was missing, that this is needed in our treatment goal or like in the therapies. And since we don't have that, and the easiest way to collect the samples can be like the microbes, we always study either the saliva or the feces. So that's like the easiest two ways, which you don't even like, that's normal. Like you can just, you don't even have to think about it as sampling. It's just a normal routine that you follow in your day. Just like give that to the lab and let them see whether it works or not. Many collaborators of our lab are doctors, but there are also like industries which will want to develop this tool, because as much as we would love to, we don't have enough funding support to develop a tool out of this. So whatever research we do, we will have to provide it to a company or like outsource the work so that they can make it a final product which can then be used by the doctors or the patients. Yeah.
I see. I see. So how expensive is it to do one of these? If it was like a kit for one person, is it easy to put that in terms of a dollar amount?
So the most expensive step of this research is the sequencing part. So it has, like, as per my knowledge, it was about a decade ago, it was more expensive than what it's now, because more and more companies are making these sequencing platforms, like machines, which are like, they will cost lesser than they used to. So because of that, and many laboratories now have their in-house sequencing machines. So that was kind of unimaginable even if 5 years ago, we wouldn't have thought about that, because that's like a lot of expensive stuff's going on in there. But now we have that, and it's like the machine is a size of the bar refrigerator that you have in your hotel room. So it's that big. It's not that big. So it's a very desktop, like, yeah, desktop machine. So it's like a very small machine. But the thing that's costly is the thing that goes in. So it's like the filters or some chemicals that we have to use to do the sequencing. So that is the most expensive, and kind of at this point where only a skilled person can do that. So, but yeah, many of the other diseases which work with microbiome, they are working towards this, where at least if you cannot do it as a kit based, I am not sure if they have that at this point, maybe somewhere in the future. But at this point, what you can do is, there are many citizen projects everywhere on the world in this microbiome. So you register online that you want to get tested for this disease, and they will send you a kit. So it's just like a bag with some tubes. So you collect your samples and you fill in the kits, and then you send it off. Like, they will instruct you how you should send it. Usually it's just like in an ice pack container, or they will, like, mostly if it's preferred if you have a contained temperature. So that's why we will request them to send back in ice packs. So after that, we receive that in the laboratories, and they will then sequence it. That sequencing may take a day or two. Yeah, 2 days. Like, it takes usually 2 days. So then the analysis will take another maybe 2 days if everybody is on the work. And then we can upload whatever we have got the results. So we can tell you, like, this much is the proportion, like, these are the chances that you have this disease, or these are the chances you have these other diseases, or how healthy or not so healthy is your saliva looking, or your feces looking. So from that, it's not directly as good as a diagnostic from the doctor yet, but you at least have the idea about what your physiology looks like. So from that point, you can approach a doctor that, okay, I got these results and I think this is something I'm worried about. So what do you have to tell me about it? So then from that point, you can have that uh, routine or like a therapy for you, which will help you attain the healthier average of the population.